Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine having a Google Maps for every single cell in your body. I mean, not just showing the major organs or even cells, but really zooming right in, seeing every tiny protein, every molecular machine, and exactly where it fits, what it's connected to. For decades, that's been, well, a huge dream for cell biologists, a true cellular atlas. And now, it seems we have the closest thing yet, thanks to this really groundbreaking study just published in Nature.
Yeah, it's quite something. This deep dive really unpacks how scientists have managed to get this unprecedented view of human cell architecture. They've mapped the location and interactions of, get this, over 5,100 proteins. And in doing that, they've pinpointed completely unexpected functions for hundreds of them. We're talking down to the nanometer scale here. It's seeing the cell in a whole new light.
Okay, let's unpack this then. Our mission today is to cut through some of that complexity, explore how these researchers actually built this cellular atlas, and maybe more importantly, what incredible insights it's already giving us about, health and disease, because this isn't just cataloging parts, is it? It's understanding how life fundamentally organizes itself.
Absolutely. And what's really fascinating, I think, is how this paper tackles that long-standing problem in cell biology. Just the sheer volume of information overload. For so long, we've had these separate snapshots, right? Different techniques looking at different things. But this team, they've created a unified view, and that lets us move beyond just knowing what is where to really understanding why it matters, how it all works together.
Okay, so before we dive into the how they did it, maybe let's set the stage a bit. Why has mapping something seemingly so fundamental, like a human cell, been such a massive challenge for so long?
Well, it's a great question. For a long time, cell biology has had some incredible tools, really powerful techniques. You've got high-resolution imaging, confocal microscopy, showing us visually where proteins are located. And then there are these sophisticated proteomics methods, like affinity purification mass spectrometry, APMS, that helps identify proteins that physically stick together, the interactors.
Right.
But here's the catch. Each of these fantastic technologies only gives you a slice of the whole picture. Think about trying to understand a big, complex city. You could look at satellite images, see the buildings, the roads.
Okay, spatial layout.
Exactly. Or you could study, say, social network data, see how people connect and interact. But you really need both, don't you? The physical structure and the connections, the interactions, to truly grasp how the city functions, how it's organized.
That makes sense.
Yeah.
So individual techniques gave us bits and pieces, like where certain proteins hang out or maybe which proteins interact. But they left these big blind spots, and crucially, you said they often led to, disagreements across different scales, from tiny machines to big organelles.
Precisely. You might get conflicting information depending on whether you were looking at the protein interaction network or the cellular location data. It's like that jigsaw puzzle analogy you mentioned. Maybe you have pieces, but fitting them together across the whole picture from the tiniest detail to the largest structure was the real hurdle. So it raised this fundamental question. How do you actually stitch all that diverse information together into one coherent, scalable reference map? That's the knowledge gap this paper is really aiming to fill. Moving from those isolated observations to a truly validated data-fused map, one that can span from nanometer scale protein complexes all the way up to micrometer scale cellular compartments. It really is like a Google Maps for the cell, and it's designed not just for exploration, but for things like interpreting disease genomes and revealing totally new biology.
It sounds like a monumental task. So, okay, where did they even begin to build this map? What kind of data did they start with? How did they start piecing it together?
Well, the foundation really lies in the sheer volume, but also the complementary nature of the data they gathered. They started with a specific cell line, U2OS osteosarcoma cells. These are pretty commonly used human cells in labs. And they systematically tagged over 2,100 specific proteins within these cells, made them trackable, basically. Then they deployed two major experimental approaches, ones that were often used separately before.
And one of those was the interaction data you mentioned, right?
Yeah,
Using
right.
APMS to figure out who sticks to whom.
Exactly. That APMS approach essentially lets you pull out a protein you're interested in, plus anything physically bound to it. This gave them this huge network, over 36,000 interactions among more than 7,500 proteins.
Wow.
And then, in parallel, they gathered immunofluorescence images, IF images. These visually show the precise subcellular locations for over 10,000 proteins. So you have the who interacts with whom and the where they live data.
Okay.
Now, the key insight, or perhaps the crucial part, was that more than 5,100 proteins were well represented in both data sets, meaning they had both interaction data and location data for this core set of proteins. And what was really fascinating, even at this early stage, was they found these two different data types actually sort of mirrored each other. Proteins similar in one tended to be similar in the other.
Ah, so that was the hint, that combining them might actually work, might reveal something more.
Precisely. It was the first indication that fusing these data sets would be incredibly powerful.
Okay, this is where it gets really interesting for me. You have these two massive, distinct data sets. One's a network map, the other's like a series of location photos. How on earth do you combine them? This sounds like you need some serious computational muscle. How do they actually fuse these two worlds?
It absolutely required serious computation. They developed this really clever self-supervised machine learning approach. Think of it like teaching a computer to simultaneously see the connections between how a protein interacts and where it lives in the cell. The AI learns the patterns in both data sets and then finds a way to represent each protein in a single unified way. It creates what they call a low-dimensional embedding.
Embedding, like a unique fingerprint.
Exactly. A unique molecular fingerprint for each protein that captures both its interaction profile and its spatial location. This allows the computer to compare proteins based on both types of information at the same time in this unified space.
Okay, so they feed in the interaction data, the imaging data, the AI does its magic. What did this combined approach actually reveal? What did the final map show them about the cell's hidden architecture?
The results were pretty staggering. The integrated map resolved 275 distinct protein assemblies. And these aren't just, small, isolated protein complexes. They span the entire scale of cell biology. We're talking tiny protein machines, maybe 10 nanometers across, all the way up to large organelles like mitochondria, which are 10,000 nanometers or 10 micrometers in diameter.
Covering the whole range.
The whole range. And crucially, the integration was key. The combined map identified 104 assemblies that you couldn't clearly see by looking at either the imaging data or the interaction data on its own.
So the synergy was real. Combining them revealed things that were invisible before.
Absolutely. It's true synergy. It provided this unprecedented lens into the cell's machinery. You're not just seeing the parts list anymore. You're seeing how they physically fit and potentially work together, like finally getting the full blueprint for a really complex engine.
Okay, 275 distinct assemblies. Many of them previously unknown, you said. How did they even begin to understand what these things were? How do you name and categorize completely new structures inside a cell?
Yeah, that's a huge challenge. How do you make sense of it all? They used a combination of human expertise and, again, artificial intelligence. They held these annotation jamborees where expert human curators would manually go through the data and try to assign names and functions based on the known proteins within each assembly.
Pretty human type.
But they also used large language models, specifically GPT-4, to help out. They prompted GPT-4 to suggest descriptive names and functional interpretations for these assemblies. And apparently, GPT-4 was surprisingly good at it. Even for the completely new undocumented assemblies, it provided confident suggestions for 104 of them.
That's wild. Using GPT-4 for biological discovery naming.
It's a powerful example of how AI can accelerate science, right? Making sense of this vast, complex data in a way that would just be incredibly slow, if not impossible, for humans alone.
It really highlights that cutting edge, doesn't it? This sort of dance between human intuition and powerful AI. So once they started categorizing, what kind of major cellular structures and compartments emerged from this big map?
Well, at the highest level, you could see the major organelles resolved really clearly. The nucleus, for instance, showed up with 102 distinct subassemblies within it.
Wow, inside the nucleus itself.
Yeah, incredible detail. The mitochondrion had 16 resolved assemblies, and then numerous components related to the cell's internal fluid and the microtubules, which are part of the cell's skeleton. They also identified 48 potential biomolecular condensates.
Condensates. These are those fascinating non-membrane blobs inside cells, right? Like little functional hotspots.
Exactly. They're super interesting because they challenge that traditional view of the cell being neatly divided by membranes. Think of them as temporary, often liquid-like droplets that concentrate specific molecules for specific tasks. And 9 of the ones they identified seem to be completely new discoveries. It really hints at a much more dynamic, maybe less rigidly structured cellular interior than we often picture.
Okay, building this huge, detailed map is one thing. But, how do you know it's actually right? How can you be sure these 275 assemblies, especially the new ones, are real? Is there a way to double-check using a completely different method?
Absolutely crucial point, and this is a real strength of the study, the validation. They used what's called an orthogonal technique, meaning something completely different from the first two methods. It's called whole cell size exclusion chromatography, coupled with mass spectrometry, or SECMS.
SECMS. Okay, how does that work?
Basically, imagine you gently break open the cells and then pass all the contents through a filter that separates things based on their size. Bigger things come out first, smaller things later. Then you use mass spectrometry to identify which proteins are present in each size fraction. The idea is if proteins are truly part of the same stable complex or assembly, they should stick together and coelute. They should travel through the filter together and come out in the same size fractions.
Okay, so if the map says proteins A, B, and C are in an assembly, SECMS should show A, B, and C coming out together.
Exactly. It's an independent physical check.
And it did work. Would they work? What did they find when they put their map to this test?
Yes, it worked remarkably well. They found significant agreement. Proteins that the machine learning approach placed in the same assemblies showed a strong tendency to coelute together in these SECMS experiments. This systematic validation provided strong support for 89 of their identified assemblies. It's like having multiple independent witnesses all confirming the same story. It dramatically increases your confidence that the map is accurate and these assemblies are biologically real.
Okay, fantastic. So we have this validated high-resolution map. What can you actually do with it? What are the big, maybe real-world questions this new Google Maps for the cell helps us finally answer?
Right. This is where the so what really comes into focus. And the researchers explored several key applications, showing the map's power across really diverse areas. Things like structural biology, understanding protein function, looking at cell type differences, even cancer research.
Let's start with the structures. How does knowing these assemblies help with understanding the 3D shapes of things?
Okay, so 3D structural modeling. They use the map, specifically the information about which proteins interact within an assembly, to guide AI-powered structure prediction tools. You've probably heard of AlphaFold. They use AlphaFold Multimer. This pairing information helped the AI predict the structures of over 1,600 pairs of proteins within these assemblies. And this led to predicting the atomic level structures for 111 protein complexes, pairs, or groups that hadn't been structurally characterized before.
So finding the partners help predict the combined shape.
Precisely. For instance, they resolved the structure of a complex called DPYSL2, DPYSL3, DPYSL4, which is important in nervous system development. They also built this large integrated structural model for a really crucial growth signaling complex called RAG regulator. And surprisingly, they found 2 new proteins, BORCS6 and ITPA, associated with it, integrating the AI predictions with existing experimental data. So it's not just identifying parts, it's getting the actual engineering blueprints, seeing how atoms fit together, which is vital for things like designing drugs.
That's incredible. Guiding AI to build new molecular blueprints. Okay, what about discovering new jobs for proteins? Did the map help there?
Definitely. It's a goldmine for revealing protein function. They started with 138 proteins in their map that had previously unknown functions, just mysteries. But by placing them confidently into specific assemblies with known roles, they could propose functions based on guilt by association. If you hang out with the RNA processing crew, you're probably involved in RNA processing.
Makes sense. Like figuring out someone's job by seeing which department they work in.
Exactly. For instance, they robustly placed this mystery protein, C18RF21, into a known complex involved in processing RNA inside mitochondria, the MRP complex. Then, crucially, they validated it experimentally. They showed that knocking down C18RF21 in cells caused similar changes in gene expression as knocking down other known members of that MRP complex. Basically, they experimentally confirmed the function predicted by the map.
So the map acts like a massive hint system for these uncharacterized proteins.
It does. And it wasn't just for unknown proteins. It also suggested new functional roles for 951 proteins that were assigned to unexpected assemblies. Proteins we thought we knew, showing up in surprising places.
Oh, interesting. Like finding an accountant working in the marketing department.
Sort of. Take DPT9, an enzyme called a serine protease. The map surprisingly placed it within the ISGF3 complex, which is involved in interferon signaling, a key part of the immune response. Nobody expected it there. Follow-up experiments then showed that if you inhibit DPP9, you actually increase the interferon response. This suggests its normal job in that complex is actually to suppress or dampen that immune signal.
Wow, a completely new regulatory role discovered just by seeing where it lives on the map.
Exactly. These are entirely new insights into how these proteins operate, driven by understanding their context within the cell's architecture.
Okay, beyond individual proteins, what about different cell types? Our bodies have hundreds of cell types. Does this map tell us if organization differs between them?
Yes, and that's a really important application. It provides a powerful platform for studying cell type specificity. They compared their map, which was built in U2OS bone cancer cells, to existing data from HEK293 kidney cells. They found about 103 assemblies that seemed conserved, present in both cell types, but strikingly, 155 assemblies showed significant differences.
So, more differences than similarities in terms of these assemblies.
In this comparison, yes. For example, they found a complex they called an energy metabolism regulation complex that was robustly present in the U2LS cells, but seemed absent or very different in the HEK293 cells. This opens up huge possibilities for identifying cell type-specific drug targets. If a crucial assembly only exists in cancer cells, maybe you can target it without harming healthy cells.
That makes perfect sense for targeted therapies.
Yeah.
Now, what about proteins that aren't just in one place? Some proteins moonlight, right? They have multiple jobs in different locations.
Absolutely. And the map shed light on this phenomenon of protein multilocalization. It was quite prevalent, actually. The map revealed that about 30% of the proteins they mapped, that's over 1,500 proteins, are present in more than one distinct assembly.
30%. That's a lot.
It is. For example, a protein called XAB2, which is well known for its role inside the nucleus, was also clearly found associated with the endomembrane system, suggesting it has jobs in multiple cellular neighborhoods. Even entire protein assemblies, like the one involving the amyloid precursor protein, famous for its connection to Alzheimer's, were clearly resolved in both the cytosol and the endomembrane compartments. This suggests these assemblies have multiple, perhaps widespread functions, challenging that simple idea of proteins having just one fixed address and one job.
Okay, fascinating. And finally, the really critical application. How does this map help us understand complex diseases like cancer?
Yeah, this is a potentially huge impact area, interpreting tumor mutations. The team looked at somatic mutations, mutations acquired during life, not inherited from 772 pediatric primary tumors. Now, if you look at individual genes, specific mutations can often be quite rare in a population of tumors. It looks like a scattershot. But when they grouped these mutations based on the protein assemblies identified in their map, the picture changed dramatically. The mutation frequency within specific assemblies went way up.
So mutations aren't random. They converge on specific functional units, specific teams of proteins.
That's exactly what it suggests. They identified 21 assemblies that were recurrently mutated across these different pediatric tumors. It indicates that mutations are hitting these functional units, these protein teams, consistently disrupting their function and likely driving tumor evolution.
That's a profound shift. Looking at the team, not just the individual player who gets hit.
Precisely. And this assembly-centric approach helped them identify 250 putative cancer proteins, proteins likely involved in cancer based on their assembly being mutated, many of which hadn't been strongly implicated as cancer drivers before. They highlighted a powerful example with the NCOR-associated transcriptional regulation assembly. By looking at mutations across the whole assembly, they found evidence implicating 3 new cancer driver genes within that assembly, which they then validated further. This could really change how we find and potentially target vulnerabilities in cancer.
This deep dive has really painted a new picture of the cell's inner world. But stepping back, beyond all these specific findings, how does this map really innovate? How does it change the game for future biological research?
Well, I think this work sets a new baseline by fundamentally unifying and extending what came before. The core innovation is that integration of multiple modes of data, the images telling you location, the interactions telling you connections. Bringing those together dramatically boosts the sensitivity and the robustness, the confidence in identifying subcellular components across all scales, from tiny to huge. They actually quantified this. About half of the new AlphaFold structures they could predict, and about 40% of the new protein functional annotations they made, were driven by assemblies that only became clear and robust when both the APMS interaction data and the imaging location data were integrated.
So that multimodal approach is really the secret sauce here.
It really seems to be the game changer. It gives you more than the sum of the parts.
And crucially, they didn't just keep this tool for themselves, right? They've made it accessible.
Yes, and that's huge for the whole field. They've released a companion cell visualization portal online. Anyone can go there, interactively explore the map, see the underlying data supporting each assembly, even look at the AI-assisted annotations. But perhaps even more importantly, they've released the Cell Mapping Toolkit. This is an open-source software framework. It allows other researchers to reproduce their entire analysis pipeline, but also to incorporate new data sets, maybe from different techniques, or to build new cell maps for different cell types or disease states.
Ah, so it's a foundation others can build on.
Exactly. It empowers the community to expand on this work, maybe integrate things like proximity labeling data or spatial transcriptomics in the future. It allows the atlas to grow and become even more comprehensive.
So looking ahead then, connecting this to the bigger picture, what's the ultimate vision here for the next, say, 5 or 10 years? How might this kind of cellular mapping really transform medicine?
I think the vision is that these kinds of cell type resolved atlases could become the standard, the default framework for interpreting things like variants of unknown significance, VUS, in clinical genomics. Those genetic changes we find in patients but don't yet understand their impact.
Right, the mystery variants.
If you can map those variants onto these detailed protein assemblies, you might finally understand which cellular machinery they disrupt. That could transform diagnostics. And similarly for drug discovery, understanding precisely which protein assemblies are malfunctioning in a specific disease and how they malfunction could lead to much more rational design of effective, targeted therapies.
It really could be transformative. Well, that wraps up our deep dive into this groundbreaking Google Maps for the cell. It's just a fantastic testament to how integrating seemingly disparate data can paint such a vivid and often surprising new picture of life at its most fundamental level.
Indeed. It really pushes us towards a new way of doing cell biology, doesn't it? Where multiple data sets are generated, integrated, validated together, all informing this unified foundational picture of the cell. And it raises a fascinating question, I think, for you to mull over. If we can now map the cell's static physical architecture in such exquisite detail, what truly revolutionary insights might we gain when we start mapping its dynamic changes? How does this map change in response to drugs, or during development, or even as we age?
That's definitely something to think about, the living, changing map inside our own cells. Thank you for joining us for another deep dive.